Now we will see the stepwise reactions of process glycolysis. It is basically categorizing or categorized in 4 stages. In stage 1, as the stage 1 has the first reaction, that is phosphorylation. So after the uptake of glucose by the cells, phosphorylation is occurred in the process of glycolysis. As the glucose is freely permeable to liver cells and insulin facilitate the uptake of glucose in skeletal muscle, cardiac muscle, diaphragm and other adipose tissue. This glycolysis is occurred in the cytosol of cells. So in first reaction, the phosphorylation after the uptake of glucose is occurred. The glucose molecule is converted to glucose 6-phosphate. A phosphate group is attached at carbon 6 in presence of enzyme hexokinase or glucokinase. The reaction which is utilized by glucokinase or hexokinase. So glucokinase is basically specific in the liver cell and the hexokinase in the liver and extrahepatic tissues, because the hexokinase is non-specific and the glucokinase is specific and present in the liver. 1 ATP is utilized and 1 ADP is formed. As adenosine triphosphate is utilized, so adenosine diphosphate is formed. So 1 phosphate group is attached at carbon 6 of glucose molecule. As this reaction is accompanied by considerable loss of energy, so this reaction is basically irreversible reaction. The glucose 6-phosphate which is formed is so important and it is associated with different pathways. It is formed from glucose and it is also reversed back to form glucose. It can lead to pyruvic acid formation by the process of glycolysis, and the pyruvic acid again go to form glucose 6-phosphate by gluconeogenesis. This phosphate, glucose 6-phosphate, can also form glycogen, that is the stored form of glycogen, by the process of glycogenesis, and it can also, the glycogen also breaking down to form glucose 6-phosphate by the degradation of glycogen. This glucose 6-phosphate also important in pentose. In this glucose 6-phosphate is also important in pentose phosphate pathway, where the 6-phosphate, this is a 6 carbon containing sugar is reduced to 5 carbon containing ribose sugar or pentose sugar. The second reaction of glycolysis is the conversion of glucose 6-phosphate to fructose 6-phosphate by utilizing the isomerase enzyme phosphohexose isomerase. The third reaction of glycolysis is the conversion of fructose 6-phosphate to fructose 1,6-biphosphate or bisphosphate, because one more phosphate group is attached to fructose molecule. And this reaction is required ATP again to form fructose 1,6-biphosphate, and this is occur in the by the enzyme phosphofructokinase, and is required magnesium ion. Again, with loss of energy, this reaction is irreversible reaction. Whereas the enzyme realize phosphofructokinase 1 is supposed to be the key enzyme in the glycolysis, which regulates the breakdown of glucose. Now in this stage 1 of glycolysis, if we calculate the energy expenditure, so in this stage 2 ATP are utilized and no ATP is formed. Now the stage 2. In stage 2 of glycolysis, the breakdown or cleavage of fructose 1,6-bisphosphate is occurred to form two molecules of triose phosphate, that is glyceraldehyde 3-phosphate and dihydroxyacetone with the presence of enzyme aldolase. So the symmetrical fructose 1,6-biphosphate is splitted down. Fructose 1,6-biphosphate is broken down or splitted down with the presence of enzyme, and two molecules of triose phosphate, 3 carbon containing sugar are formed, and aldotriose glyceraldehyde 3-phosphate, and another one is ketotriose, that is dihydroxyacetone phosphate. This reaction is reversible, can reverse back, and in this stage you saw that no energy either utilized or formed. The two trios are interconvertible and only the glyceryl 3-phosphate is able to undergo further process of glycolysis. In glycolysis, the dihydroacetone phosphate is rapidly converted to glycerol 3-phosphate. So two molecules of glycerol 3-phosphate are formed. This reaction can be inhibited by bromohydroxyacetone. Why? Because it resembles to dihydroacetone phosphate, so can bind to enzyme phosphotriose isomerase and can stop the reaction. So by stopping the reaction, accumulation of dihydroacetone and fructose 1,6-biphosphate is occurred, and the further reactions of glycolysis are stopped. Now, stage 3 of glycolysis. This stage is basically energy yielding stage, and potentially useful energy is formed in this stage. The first reaction of the stage is oxidation of glyceride 3-phosphate to 1,3-biphosphoglyceride. Means two phosphate groups are attached. As the dihydroxyacetone phosphate is also convertible or converted to glyceride 3-phosphate, so both are involved in the formation of 1,3-biphosphoglycerate, and this required coenzyme NAD. If the case is aerobic glycolysis, so the NAD is the NADH which is formed in this reaction is converted again to NAD, and mitochondrial oxidation is occurred with the liberation of 6 ATP, and this is 6 because of one NADH production, and oxidation of this one NADH liberate 3 ATP, so total 6 ATP are formed in this reaction. The second reaction of this stage is conversion of 1,3-biphosphoglycerate to 3-phosphoglycerate. This reaction is occur in presence of magnesium ion, and 1 ATP for each 1,3-biphosphoglycerate is formed. So 2 ATP are formed in this reaction, and simply the phosphate group is going to attach with ADP, adenosine diphosphate, to make it adenosine triphosphate. The importance of this reaction. This is basically the unique example where the ATP is produced at substrate level without participating in electron transport chain. So what is this substrate level ATP production? In this type, basically ATP is formed at a substrate level. And that is why it is called substrate-led phosphorylation. And this is the type of reaction where ATP is not participating in electron transport chain, and thus it is called substrate level phosphorylation. It is important to know how the reaction can be inhibited. The arsenite, if present, it competes with inorganic phosphate. So in the reaction of conversion of glyceride 3-phosphate to 1,3-biphosphoglycerate, and it produces then 1-arsino-3-phosphoglycerate, which is then hydrolyzes spontaneously to yield 3-phosphoglycerate and the heat, and thus reaction is inhibited. And thus in this step also no ATP is formed. Secondly, iodoacetate and iodoacetamide can also inhibit the reaction. How? They bind covalently with acid group or alkylate the acid group of the enzyme. What enzyme? Glyceride 3-phosphate dehydrogenase. And because of this, accumulation of glyceride 3-phosphate occur, and the process is inhibited. If we calculate the energy production at this stage, so 6 ATP total are formed from the oxidation of NADH if the case is aerobic glycolysis, and also 2 ATP are formed at substrate level. So total 8 ATP production occurred up till now in this stage. As in first, ATP are used. So the stage 4 is basically the recovery of that phosphate group. How? From 3-phosphoglycerate. The 2 molecules of 3-phosphoglycerate, the end product of the previous stage, which is formed, is still retain the phosphate group. So it is the stage where recovery of ATPs occur. How it is occurred? First, the reaction in first reaction only the rearrangement of phosphate groups occur. Conversion where the conversion of 3-phosphoglycerate to 2-phosphoglycerate is occur in presence of enzyme phosphoglycerate mutase, and 2-phosphoglycerate is formed. Now in second reaction, conversion of 2-phosphoglycerate to phosphoenol pyruvate by dehydration is occurred in the presence of enzyme enolase. This enzyme is dependent on magnesium or magnesium. The process of glycolysis inhibited by fluoride ion at that state. Now, as I told you that it is the stage where recovery of phosphate occurs. So, phosphoenol pyruvate is then converted to enol pyruvate. This reaction is catalyzed by the presence of the enzyme pyruvate kinase, and the high energy phosphate of phosphoenol pyruvate is now transferred to adenosine diphosphate and thus liberating adenosine triphosphate or ATP is recovered. And as the two molecules are there, so 2 ATP are formed. And this is also magnesium dependent enzyme which is involved in this reaction, and the reaction is irreversible, and the energy production is also again at the substrate level where electron transport chain is not involved. The enol pyruvate which is formed is spontaneously convertible to ketopyruvate. If you talk about the energy production at that stage, so 2 molecules of ATP are produced as 2 molecules are involved in this reaction. So per molecule of glucose will produce 2 ATP in this reaction, which is at substrate level. We can also calculate the energy throughout the reaction, which is up till now taking place. First, at first stage, 2 ATP are utilized, and in second stage, stage 2, there was no production or neither utilization of ATP, whereas in stage 3, 6 ATP are formed via mitochondrial oxidation, and also at substrate level 2 ATP also formed. The total 8 ATP are formed, and in stage 4 2 ATP are formed. As the 2 ATP are consumed in first stage, the total energy or total ATP recovered or net gain of ATP are 8. We can also represent the overall mechanism in a equation form, say that 1 glucose, 2 NAD are utilized, 2 ADP are utilized, 2 phosphate group utilized, and producing 2 pyruvate molecules, and with 2 NADH, 2 ATP and 2 water molecules, and that 2 NADH further produce 6 ATP. So when we calculate, so we can easily come at the level that total 8 ATP are formed up till pyruvate synthesis. And now this all process which is occurred in cytosol of the cell can goes toward mitochondria, where further process occur if the oxygen is present. Now in case of anaerobic phase of glycolysis, how much energy is produced and why not produced as comparative to aerobic phase of glycolysis? As I already told you, the reoxidation of NADH is required oxygen, or mitochondrial oxidation is not there. So it cannot take part in electron transport chain, and thus cell as usually probably have limited coenzyme NAD. So to continue the glycolytic cycle, NADH must be oxidized to NAD. but this cannot take part in electron transport chain. So what happened? It is achieved by the conversion of pyric acid to lactic acid. But though ATP is formed, it is involved, or the enzyme involved in this reaction is enzyme lactate dehydrogenase, and this is the reaction which is irreversible. I already told you, when the oxygen becomes available, so again pyric acid is formed. Now see the reaction. The ketopyruvate which is formed in previous reaction, it can be converted to lactic acid if oxygen is not present, or in the anaerobic phase of glycolysis in presence of lactate dehydrogenase, and that recovered the NAD, but no ATP production is there because no electron transport chain or no availability of oxygen. Dehydrogenase can be inhibited if oximate is present, as we are discussing the inhibitor with the reactions. So here the oximate is responsible to inhibit the enzyme lactate dehydrogenase. So the reoxidation at this process also inhibited, and the process of glycolysis under an anaerobic phase is stopped. If you summarize the anaerobic phase in a reaction form, so only 2 ATP are formed, because overall 4 ATP are formed, whereas at first stage 2 ATPs are consumed, and no 6 ATP which were formed in aerobic phase, and they are produced because of mitochondrial oxidation, which is not present in case of anaerobic phase of glycolysis. So one glucose molecule, 2 coenzyme of NAD, and 2 adenosine diphosphate, 2 phosphate group will lead to formation of 2 lactic acid molecules and only 2 ATP with liberation of 2 molecules of water. That is basically the difference in the reaction as compared to aerobic phase of glycolysis. Also summarizing, I am repeating the things to make you more clear again and again. So hopefully you are getting my point. Simple, the reaction is same up till 2 glyceraldehyde 3-phosphate, and also at this stage the second compound 1,3-diphosphoglycerate are formed, but no presence of there is no availability of oxygen. So 6 ATP are not formed at that stage, and at this stage where the pyruvate is converted to lactate, this provides the recovery of coenzyme NAD. So total 4 ATP you can see here are formed, while 2 ATP in the first stage in 2 reactions are consumed. That is why only 2 ATP formation in anaerobic phase of glycolysis is occurred. This anaerobic reaction is also important. See some clinical importance. As tissue also perform their action under hypoxic circumstances. We have certain examples, and they at this stage produce lactic acid from glucose oxidation and producing local acidosis, and also if the lactate production is more, it can produce metabolic acidosis because no oxygen is there now. Accumulation in the skeletal muscle will produce relative anaerobiosis, and the glycolysis will produce lactic acid, and the lactic acid accumulated in the muscles will cause changes and thus produce fatigue. The accumulation of lactic acid in the skeletal muscle, which can be increased form and also cause or increases the permeability of muscles, capillaries, and causing edema, the interstitial edema, because they come in the interstitium, and also white blood cell infiltration. That is why the reaction, or it has clinical importance to know about this lactic acid formation. And this is, however, reversible. Whenever oxygen is available, it's again converted to pyruvic acid, especially our white blood cell functioning under certain circumstances where oxygen is unavailable somehow. So this anaerobic phase is proceeding. As a small amount of lactic acid are produced from pyruvic acid, even in resting condition. As I told you, the best example is erythrocyte, which derive even 90% of their energy from glycolysis, and other tissues such as which produce small amount of lactic acid. They include brain, retina, and skin cells. So the anaerobic glycolysis has it's impossible too, and the process is not stopped. So that is why in aerobic phase the glycolysis also continue but produce lactic acid. This lactic acid after formation can come in the blood, and 20 to 30% of it undergo complete oxidation in various tissue and through citric acid cycle, but require recovery. They require further reconversion to pyruvic acid, while the remaining part goes to the liver to form glucose by gluconeogenesis after the conversion to pyruvic acid. Means both these processes require conversion of lactic acid again to pyruvic acid in presence of oxygen. When the oxygen become available, so they again converted to pyruvic acid. It is increased, this reaction or unavailability of oxygen is increased. So relative anaerobiosis occur and glycolysis will stop as the cell will exhaust, or NAD is going to be finished. So at a certain time, the processes continue, and if oxygen become available, so reconversion of pyruvic acid occur, and if it is not present, then after a certain time, glycolysis will stop as the cell will exhaust coenzyme NAD.